Hi again. Good evening. I'm Dr. Philip McMillan. And for those who don't know me, I've been focused on autoimmunity around COVID 19 since March 2020. I think it's an essential part of understanding the science of the disease and why severe disease seems like such an unusual pattern. And so part of my role is to share scientific insights because I continue to research COVID 19 and long COVID and look at the angles around the disease. So today I'm talking about this what is considered to be misinformation. Is mRNA vaccine shedding possible? I was in two minds when I thought about talking about this because I'm not trying to be controversial here, but my primary aim is to target the question about masks. For some reason, we have been caught in an argument about whether or not masks are effective. And there is some research that shows it makes no difference. Some research shows that it may make slight amount of difference. The principles are very simple in terms of science. If there's anything that you can do to reduce the risk of infection, it's worth doing. It would be like arguing about hand washing. It doesn't stop an infection, but the small actions can make a difference. And so, specifically, one of the target principles is that many people are very anti mask because they suspect that the viral particles are so small that they would get through the mask anyway. And I understand that principle, but the truth is that anything that spreads is likely to be in salivary droplets. And that's what I'm going to be demonstrating here today. And so it brings me right to the central bit of the discussion about the possibility of mRNA vaccine shedding. Now, it's important to note that vaccine shedding is usually only related to live virus vaccines. Because in some cases, where you do a live virus that has been altered so it doesn't cause a severe an infection, that virus, in some cases, like for instance, I think in the measles vaccines and the live ones, can then be spread. And that's what they consider to be vaccine shedding. So the misinformation that they argued about, or the fact checkers said, was that mRNA is not a live vaccine, so it is not possible for it to be shed. And I agree with that scientific perspective, but I think that is missing the point. And that's why I'm focused on this principle about can RNA vaccines be shed. So, in order to understand it, I'd have to encourage you to visit my Substack. And the reason for that is because I have some important points in my Substack about effective mucosal immunity. And this is essentially why I'm talking about the possibility of mRNA vaccine shedding, because more and more evidence is coming out that natural immunity does seem to be superior to hybrid or vaccine mediated immunity. So, if you do have natural immunity, you would want to protect it. And this is part of the discussion about could we end up with the mRNA nanoparticles impacting on natural immunity? And that's the scientific question that I have. So, this is not about a research point. This is purely a theoretical look at some of the challenges that we have. So I go back to the main picture here and I'll bring it on full screen. And essentially, you have here someone who is coughing or sneezing. I've put a few viruses in there, but in a sense, I'm not really focused on that. And you can see here, these are the viral particles. But critically, when someone coughs or sneezes, or even breathes or talks loudly, the salivary droplets are spread. And it's salivary droplets that really carry the risk rather than a single virus floating on its own. And it's these salivary droplets that I think are relevant in terms of the question about mRNA vaccine shedding. So here are the first steps. The first question is, when mRNA is injected into the muscle, does it stay there? Now, the research indicated that it did. That was what we were told initially, that the mRNA particles would be taken up by muscle cells, and there will only be spike protein produced, which will create an antibody response, but the mRNA particles themselves only stay in the muscle. So that was purely an idea rather than fact, because when it was actually studied, and I will show you here, do mRNA vaccine particles circulate? This is what I did in July of this year on Substack. And yes, the evidence came out from the paper here, and this paper was done in Romania. And what it had highlighted is that vaccine mRNA can be detected in the blood at 15 days post vaccination. And so it's an important piece of published work. It was published in June 2022. So this is only relatively recent. The importance of it was that it was the first time we conclusively demonstrated that no, the mRNA doesn't just stay in the muscle. It can actually circulate in the bloodstream, and in that case, for up to 2 weeks. So the next question that was asked was, could the mRNA get in any kind of secretions? And yes, it was demonstrated it could get into breast milk for a short period of time. It could get into saliva, again, for a relatively short period of time, depending on the level of research that was done. My view is that if it's in the bloodstream, it certainly, even small amounts, could end up in saliva. And this is where that question about mRNA vaccine shedding occurring, whether or not it's possible. So I did a few images here in BioRender to help to clarify the point. As I pointed out to you in this image here, again, I make it, each of the droplets is a collection of liquid with either saliva, well, this is the saliva with probably some proteins, water, and any other thing that is in the saliva. In that context, I've taken one droplet here, and I've used this as a template, and I've created this in BioRender to show you. And inside this droplet, there are multiple other little droplets all coming together as one thing. And within this single droplet here, you can have multiple, potentially, mRNA particles. And therefore, that mRNA particle could potentially, therefore, be spread from person to person. And this idea is not strange because they have discussed and tested the idea of self-spreading vaccines. And this was used in animals and demonstrated, just hasn't yet been accepted as being safe enough in humans, where you vaccinate one cohort and they spread, I guess, a viral vaccine to other people and therefore get herd immunity. So the idea is there. It is scientifically feasible. And the question is this possibly occurring even in our day-to-day existence? If someone has recently been vaccinated or boosted and they may have high levels of circulating nanoparticles, could that be spread to someone else who either has been vaccinated before or not? And therefore, could the lipid nanoparticles then be absorbed into the cell? And this is what I have here. So a lipid nanoparticle, if it got into the nose or mouth, would then go through the cell wall and then release the mRNA to make spike proteins, which would then trigger an immune response. And this is the image of a typical lipid nanoparticle. This is the Moderna one. The Pfizer one is very similar, and it has this lipid outer ring, and inside is the mRNA. So it's housed quite carefully in there. Here is an important differentiation. Although they found mRNA in the bloodstream, my suspicion is that it's not actually circulating lipid nanoparticles, because the lipid nanoparticles are quite fragile, so they usually break down. So the context that I have, the only thing that makes sense to me is that the body has a natural system to circulate information proteins called exosomes. And exosomes, this is a picture of a cell. This is taken from a review of biophysics and biology. And what it shows you here is that cells, most cells will package little proteins, put them together in a lipid nanoparticle, and then spread them through the bloodstream to different parts of the body. And these are exosomes. So it seems almost logical that even if a viral RNA, mRNA from the vaccine comes into a cell, a portion of it may become packaged into exosomes and then spread in the bloodstream. And that's the reason why I think it's not actually lipid nanoparticles that they found in Romania when they looked at the research, but they may be exosomes that contain mRNA. But the principle is still the same, is that if it can get in the bloodstream and in breast milk and therefore in saliva, it is possible that it could then be spread by coughing, sneezing, or even someone speaking loudly. That's the most important point that I want to get across. At the end of the day, a mask is more likely to stop a droplet. And droplets are usually what have the ability to travel longer distances. And so, my point today is to encourage everyone, whether or not you are pro vaccine or whether you are against the COVID vaccines, that masking seems to be the most simple and effective strategy that we can use as we head into a difficult winter. Remember, we're seeing what's starting to happen in China, and they wear masks all the time. And so, even within that, it will still be spread. We're just talking about reduction of spread. To me, it makes perfect sense from a scientific point of view that everyone, as best as possible, should try and use masks whenever they can. We use it all the time at work. We've become quite adjusted to it. And so, therefore, if we can do it in the clinical context around patients and around each other, there's no reason why anyone else can't do this in their day to day activities. So, I am using the principle of mRNA vaccine shedding to reinforce the point that as we go into this winter, we need to, as best as possible, for those who may be unvaccinated, protect their upper airway because the natural immunity you may have would be more effective potentially than spike mediated immunity. So protect that. And in the same breath, you are also protecting yourself from a viral infection or reducing the risk both of flu, RSV, and COVID. And so that should be a valuable thing for all of us to do. So I want to wish everyone a Merry Christmas. Try and stay safe. And I look forward to sharing more ideas with you about the science of COVID-19 in the near future. Have a great evening.